So often when we talk about something complicated, it's useful to simplify the narrative. So I'm going to simplify this narrative by focusing on one particular species, salmon, and particularly coho salmon. So coho salmon is an excellent sentinel for stormwater effects for a variety of reasons. Many of you probably know that they're widely distributed. This is Puget Sound, and what I'm showing here are lowland streams that coho tend to favor. So they tend to be found in these lowland streams, which overlaps with where humans like to have their settlements as well. So we have a lot of potential to overlap and impact coho salmon. They spend about half of their life in fresh water. So that's really important because we can control contaminants perhaps before they enter Puget Sound if we can focus on coho and making sure they are healthy. They're supported by a diverse food web. They need a good amount and quality of water in order to support that food web and then therefore themselves. And of course we also focus on coho salmon because they are listed as a species of concern in Puget Sound, and they're listed as threatened in other regions due to a variety of factors. So Pacific salmon more generally are in decline throughout their natural habitat. This shows an image from one of NOAA's websites where habitat typically utilized by salmon is have populations that are either listed on the Endangered Species Act as threatened or species of concern. Now we know that this is our fault. We're pretty sure this is our fault. And there's a variety of factors besides what happens in the ocean. If we just focus on freshwater, we know that dams can be a problem. We know that logging practices can contribute to these declines. We know that hatchery practices can contribute to salmon declines. And then there's also the many insults of urbanization, which is one, as Martha said, the death by 1,000 cuts, or 1,000,000, as she said. Now, one factor of urbanization, of course, is impervious surface area. And this is a map for across the United States. And you can see some hot spots where there are lots of impervious surfaces, where the water can't seep into the soil. But impervious surfaces are kind of like fractals. If you focus in on a region, you find the same patterns again and again on smaller and smaller scales. So it's not just an issue for really highly built out areas. Anywhere that you have impervious areas, you're going to start running into ecological problems. This figure here is just showing, I don't think I have a pointer on this. Does anybody have a pointer? There's one up here somewhere? Ah, right here?
Right there.
All right, let's see if I can figure that out. Oh yeah, all right, she's smart enough. All right, so impervious area here, and then as you increase impervious area, you get a decline in this benthic, what's this called, inventory of biotic?
Index.
Index of biotic integrity, which is just one measure of qualifying how healthy an aquatic habitat is, looking at benthic invertebrates. You can see that there's no threshold here, there's no safe area, and then it starts to decline. Pretty much as soon as you start introducing impervious habitat, you're going to have a decline in habitat quality. That this is a problem is because this is not a forest, and because it's not a forest, it doesn't act like a forest when it rains. Water rushes quickly off of these surfaces, and it carries with it any contaminants that are sitting on top of them. Sometimes they also generate contaminants. Does that pollution affect salmon? It's a little hard to get to those answers because there's so many factors happening at the same time. Stormwater is a very complex mixture of contaminants. So we have metals, we have pesticides, we have surfactants, lots of things are involved, and a lot of them are coming from vehicles, sometimes building materials, landscape practices. Recently, a Department of Ecology, Washington State Department of Ecology report pointed out that the greatest, and I'm sorry you can't see all of these, I'll try to point out what's at the bottom that you can't see here. The greatest opportunity for toxic chemicals to be transported to Puget Sound and its freshwaters occurs during storm events. So this is really important that we're starting to focus on storm-related contaminant transport. You know, decades past, we had big point source pollution, and we've done a pretty good job of eliminating the larger point source pollutions, and now it's more these many small inputs that we're all contributing to. So as I said, I'm going to be focusing on salmon, and salmon have a higher risk for impacts from stormwater runoff when they're in fresh water. So here we have adult salmon that are returning to freshwater to spawn. They lay their eggs, the eggs hatch and develop, in the case of Coho salmon, for another year after the hatch. And then they head to the ocean, grow, and return as adults. So I'm going to tell a few stories here of stormwater impacts on certain life stages of Coho salmon. So the first story is stormwater affecting adult coho when they return to spawn. So throughout Puget Sound and a lot of other areas where salmon are also experiencing declines, local municipalities, City of Seattle, for a lot of the stuff that I'm showing here today, has spent a lot of time and money restoring urban creeks, with the hope that if you rebuild it, the salmon will come. So they did a really wonderful job. Here's an example. I think this is Longfellow Creek here in West Seattle. Really fabulous job restoring the physical habitat. You know, these systems look pretty good now. There's ponds and ripples, and the sinuosity, the curving of the creek has been reestablished. So after this work was done, they started sending out survey crews to, well, did it work? Are the salmon utilizing this habitat? And in fact, yes, they found adult salmon had returned to these systems to spawn. Very exciting. Unfortunately, they found that the salmon were overall dying before they had a chance to spawn. And this is not normal. This is a trace of a variety of things for this Longfellow Creek in West Seattle. The first year that NOAA Fisheries was involved in daily spawner surveys. They go out every single day, and here's the rainfall. This is one of these dry summers. The rain started relatively late here, beginning of November. Here's the creek flow, which tends to match the rainfall patterns because it's one of these very flashy systems. A lot of impervious area. So we started to see coho showing up in the stream after this first fall freshet. That's what draws them up. The water raises up, it gets high enough they can get in, it's cold enough, they feel it's right. And as soon as fish started showing up, we would find them dead. And it wasn't until later in the season that we started to see some spawned out carcasses as well. I have a few videos to show what this phenomenon looks like. So here's an adult salmon, and he's still, she probably in this case, so still silver, you know, hasn't even changed into spawning color, so it's recently entered into fresh water, and it's dying prematurely. It's displaying some very uncharacteristic behaviors, this splaying of the fins, gaping, otherwise a very healthy looking fish. There's another example from this one from Longfellow Creek. Often these salmon will be disoriented, and their behavior is very abnormal. Usually adult salmon are a little shy, and if they're not yet in full mating behavior. So this one's disoriented. If you're in the creek, they'll just swim right by you, bump into your legs. Here's another salmon that just, this loss of equilibrium associated with it. And in this final video, which some of you may not be able to see, we're looking at a coho salmon underwater. Again, these silver bright colors. And just upstream are, this particular year, there's a really good overlap in the chum spawning run. So adult chum are actively spawning right upstream of where this coho is gasping and dying prematurely. This is part of that downer phase. But it's real and it's happening right now, and so I think it's really important to raise awareness about it. Here's some images of some of the females that we found over the years in these studies, different systems, different years, just chock full of eggs. Some are less mature than others. And across the streams where we've studied this phenomenon, we found pre-spawn mortality rates of 60 to up to 90%, 100% sometimes of the run in years when there weren't many salmon. But even in years where there are a lot of salmon, it can be 70, 80, even 90% of the run dying prematurely. So we spent a lot of time over the years doing forensics, trying to understand why this was happening. Unfortunately, there's no smoking gun. Regular conventional metrics like dissolved oxygen is fine, temperature is fine. There's no one contaminant that's above some toxic threshold that we're aware of. We know that coho are exposed to contaminants like PAHs, which we know can be toxic. So here I'm showing you, this is phenanthrene and benzoylpyrene. These are two different PAHs. And I'm going to have to explain it because the screen's a little low here. So this black bar is Longfellow Creek, which is that urban impacted stream. And then we have Fortin Creek, which is a really pristine system. You can see that the pH levels were much higher than in the pristine system. In another year, again, we're seeing much higher levels in the urban creek than in either Elliott Bay before they enter the freshwater system, or in this case, it was a rural hatchery. So they're obviously exposed to pHs, and pHs can cause cardiovascular toxicity. Is this what's causing the mortality? We're not sure. We also know that they're exposed to metals when they enter these urban freshwater environments. Higher levels, you can't see it here, but this is cadmium, lead, and nickel. Higher levels were found in the two urban systems compared to the pristine system. So metals can be acutely lethal. They cause osmoregulatory problems for fish. Basically, it causes them to suffocate. Is this what's happening? The levels of those metals were not above any given threshold. It may be that adult coho are an especially sensitive life history stage. Most of these toxicity studies were done on smaller juvenile fish. Adults, we know, are suffering a lot of changes in their bodies. They're literally falling apart as they mature. So it could be that they're just more sensitive at that stage of their life. And further studies would be good to identify if that's what's going on. What we have been able to tell, though, is that this phenomenon is associated with certain land use types. So there's a large study that came out, again, you can't see it unfortunately, but Feist et al. Came out last year in Public Library of Science, which is an online accessible journal. And a big GIS analysis showed that basically this pre-spawn mortality phenomenon is associated with, no big surprise, but you could associate it with impervious surfaces, roads that are heavily used, commercial roads, densely urban areas. Not too surprising, but important as a first step towards understanding where else we might be finding this happening. Something else I wanted to show today, even though it's a little complex with the graph I'm going to show up in a minute here, but I think it's really important that we understand. If we do, it's not very difficult to put together some models that look at where this is happening and what will happen to the larger population. Because usually salmon populations aren't isolated. You don't just have one population. Usually it's more like this, where you have a stream, a main stem, and then there are creeks jutting off of that, which have their own little salmon populations. So what happens, for example, if one of these populations is impacted by pre-spawn mortality? Perhaps it's development. Let's say it's development. You develop that watershed, suddenly you're seeing this premature mortality. Well, basically what's going to happen, here's assuming there's no pre-spawn mortality. Now you add it to one of the basins. Well, that one population is going to suffer decline, but the others stay fairly high with a little bit of decline. Let's say another basin gets developed and you'll see pre-spawn mortality there. Well, the second population declines, but what starts to be obvious is that overall the populations will start declining as well. So let's add more basins here, more basins. You can see that there's still relatively sustainable populations in the unharmed, the untouched basin. And it's not until all of the basins are impacted that you'll have these populations all head towards extinction. So what's the take-home message from this exercise is that pristine populations help support impacted populations. So it's really important that we maintain at least some pure pristine basins where you can have these source populations to recolonize impacted basins. To a different life cycle stage, an example of stormwater impacts on coho salmon at the embryo development stage. And I am going to be talking about cardiac toxicity stuff with salmon, so brace yourselves. This is a study that was done a few years ago now, again at this Longfellow Creek research station. And what we did was we tried to clean up the water. So we don't have a control, pristine mountain stream water. We have the urban creek water from Longfellow Creek, unfiltered, which looks like this, and then filtered, try to clean it up as much as possible. And this was a combination of sand filtration, activated charcoal, and carbon filtration. And what we did was we took these trays of developing salmon embryos, coho salmon embryos, and exposed it to these two different water types. Wait and see what happens. So we allowed the coho to develop about 50 days, and this looks a little mangled because the yolk sac has been removed from these embryos. But in the unfiltered water, the embryo looks fairly normal. As a developmental biologist, you would understand this looks like a normal developing embryo. However, the coho that were in that unfiltered water, just the straight stream water that was running alongside, do not look normal. They're developmentally delayed, they're much smaller, and they have these pockets of cranial hemorrhaging. They actually had blood spots in their heads. I have another figure here that shows a different view. So this is a top-down view of the embryo's head, and you may be able to see here these patterns of the capillaries in the head region. And in the unfiltered water, you can perhaps see that that pattern is not, the beautiful, perfect pattern is not there. In fact, the vasculature is completely disturbed with, again, just some blood spots. What I'm showing here in this figure is over time, again, it may be hard to see, this is 35 days post-fertilization, so they've been developing for 35 days, and we go up to 62 days of development over here. And in the filtered water, we're seeing a low percentage of abnormal embryos. But in the unfiltered water, the regular stream water, the percent of abnormal embryos goes up over time. So by the end, you've got almost 80% abnormal, which included dead embryos also that had these cardiovascular defects. All right, so that's the really depressing stuff. Enter the promise of green stormwater infrastructure. So I'm sure you've heard about these. I'm sure you have some here on the island. They include things like bioretention systems, green roofs, permeable pavement, which can be a really stark demonstration when you see one juxtaposed with regular pavement. So the idea is that some of these technologies can help reduce or prevent impacts from stormwater. Now, a study that I'm involved with currently is a big collaborative effort, and I just want to impress upon you how important it is these days that we have, or that we can pool resources from different areas of science. So, State University, the Washington Stormwater Center is part of it is located there, and that's who I'm doing this work with. U.S. Fish and Wildlife is involved in this program. NOAA Fisheries, particularly their Coastal Storms program, and EPA has supported a lot of this research as well. And here's what we're doing. We're focusing on the acute impacts that are happening over short periods of time from exposure to stormwater contaminants. And first, what we're doing is determining, okay, what's in this water? If you take something like a highway runoff, what's in there? And then evaluating what the toxicology baseline looks like if you, being a fish or invertebrate, have been exposed to that water. And then where we're heading with this is the next step will be to identify which of these green stormwater technologies can minimize the direct impacts of salmon and the indirect effects that happen through impacts on the salmon food web. So when I'm talking about acute impacts, one thing I want to point out is that this idea of first flush of contaminants during a storm. So this is storm duration as the storm goes on, and then concentration of some contaminant, contaminant X. And it tends to be focused at the beginning of a storm. So when water first starts washing an impervious surface, it's going to collect those contaminants and then move them to wherever you're concerned about them going. In contrast, so if we're concerned about acute impacts, we're concerned about what's happening during this first part of the storm. In contrast, a lot of studies that have looked at what's in stormwater will look at a flow-weighted mean. So this stands for event mean concentration, and they'll just collect water across the entire storm and then determine what they found. And at that point, you're looking at a much lower concentration of whatever contaminant might be there early on in the storm to cause an acute effect. Here's an example from the literature. This is a highway storm. I think this was a Chinese example, but really this is a pervasive problem across anywhere that we have impervious surfaces in developing areas. I hope this is gonna stand up. Okay, so lots of things going on here, but I've highlighted the important things, which include here in blue the flow rate during the storm. So basically, it starts raining and then the water starts flowing and then it tapers off towards the end of the storm. And during that same time, an example of metal mobilization here denoted by PB for lead, and also the PAH mobilization, which is the organic hydrocarbons. You can see that it's a very similar pattern to what I showed with the cartoon over here. So from the little bit of literature that there is on the actual toxicity to biology of stormwater runoff, you tend to find that most of the toxicity is in fact associated with that first part of the storm. And the primary suspects for these toxic effects tend to be metals and PAHs. All right, so what I've done with this work so far in order to characterize the water chemistry and assess the baseline toxicity was to utilize an urban highway that was very accessible to me, because 520 dumps its downspouts right into the parking lot at NOAA Fisheries. Yeah, very convenient. So here's the downspout, and here's an example of some first flush runoff that I collected for one of my earlier tests. Yeah, it's quite fascinating to be up close and personal with it like that. It tends to be darkly colored. It tends to smell like a variety of things. Sometimes it's a little poopy smelling, sometimes it smells like an ashtray, and obviously each event is going to be slightly different, and that's part of the challenge of working with complex mixtures. Some of the tools that we brought to bear on this problem include being able to use invertebrate and fish surrogates for salmon and their food webs. So what we wanted to do was to use these high throughput species to start to identify, okay, what are the problems? What does happen to the biology without having to wait a year for a salmon to grow up and be able to assess it in that way? And some of the salmon food web is difficult to find or ephemeral, so these are really useful tools for us. With the invertebrates, I was able to look at a lethality test and also a growth and impairment test. And with zebrafish, there's a suite of tools, including morphological disturbances, cardiac toxicity, and mechanosensory. Well, I'm really getting into that, so I won't bother explaining it right now. All right, so we take some of that runoff and we expose it to the invertebrates. And here's an example just to show that some of these storm events can be 100% lethal. So here I'm looking at survival of the invertebrate over a 2-day test, which is a pretty standard toxicity test. 100% lethal in basically the fresh runoff, but runoff that was stored in the dark in cold storage for a week was rendered, I wouldn't say non-lethal necessarily, non-toxic, but it was non-lethal, which is interesting and suggestive. I'll get into that a little bit later. The reproductive test is also a fairly simple test. Basically, we take some of the runoff, we add 10 of these little baby bugs, allow them they start to reproduce, and after they've had a couple cycles of that, then we take them all out and count them. So it's a pretty crude test, but very telling. So here I'm looking at relative abundance. I think this is backwards done. Relative abundance. So a value of 1 means it's the same as the controls, okay? So here I'm showing different storm events. I wish you could see it, but these are 5 different storm events, looking at the number of females that were alive at the end of the test. Here they're all dead. Here they're all dead. Here they're mostly dead. These ones, statistically, you could say, okay, nobody died, as far as statistics tells us. But offspring for a female tells us about their reproductive success during this time. Obviously, if they're all dead, then they had poor reproductive success as well. But among the ones where maybe statistically nobody died, as far as the initial individuals you put in there, you can see that their reproductive success rates were incredibly low. And even here, just as far as relative to the number that are alive, they're not reproducing well at all. To the invertebrates, and among the non-lethal storms, reproduction was always impaired. For the fish side, I use zebrafish, which is a really popular aquatic lab rat these days. And one of the reasons is that it's very popular in medical research, so we know a lot about this animal. Toxicologically, they're very similar to mammals. They're optically clear, which makes them really easy to study because you can literally watch them develop. You can watch their organs develop. You can watch the blood flowing through their blood vessels. They develop very quickly, and probably because of their use in medical research, their genome's been completely sequenced, and there's really great potential for genetic biomarkers. So here's an example of their developmental sequence. Their heart starts beating about 24 hours. They hatch out about 2 days, inflate their swim bladder about 3 days, and then you're pretty much looked at most of the important developmental sequence. So some of these same runoff events that I just showed for the bugs, here we're looking at what's a 4-day test for fish, and you can see that some of the storm events are also 100% lethal to the fish. And these tended to be when this ADP stands for antecedent dry period, which is basically the number of days before it rained. The number of days it didn't rain before it did rain. So for storms that it was dry for at least 2 weeks, those were very lethal to the fish. And here's an example, an image example from one of those storms. So the top line here is controls for each of those 4 days. My pointer's not working so well, but 24 hours, they have this beautiful shape here. 48 hours, they are perfect and they start to hatch out. And then by the end of the test, they've got their inflated swim bladder and they're these lovely, beautiful creatures. The bottom is the 4 days for fish that were exposed to that runoff, the lethal runoff event, and the orange bars at the bottom are showing cumulative mortality. So by the end of the test here, everybody had died in this experiment, but the survivors along the way never continued to develop, which was, as a toxicologist, pretty interesting, but disturbing as well. Some of the more subtle effects that I would see across storm events were things like this, delayed hatching. So even down at the numbers I'm showing here on the bottom, these R's are dilutions of the storm. So R100 on the far right is just the straight runoff. But even down at a 3% dilution, so that's 3 parts of the runoff to 97 parts clean fresh water, there's a delay in their ability to hatch. Another sensitive and reliable effect that I would see was an inability to inflate their swim bladder. So that's what allows fish to have neutral buoyancy, and they save a lot of energy by having a swim bladder instead of having to swim around all the time. And I was seeing this particular storm event, we see even at 12% dilution, a lack of inflation of their swim bladders. And this didn't get better when I transferred them to fresh water. So this was a fairly permanent effect. But those effects, delayed hatching and inflation of your swim bladder for a zebrafish, that's not a salmon, that sort of the so what question is a little confusing. Some of the effects that I've seen that are a little more reliable as indicators of potential harm to any fish species, things that we understand a little bit better, include cardiotoxic effects. So here I'm looking at different dilutions of a storm, and this is one of these storm events that was not lethal. There was in fact no mortality, but pericardial area is what I'm measuring here. And what that is, here's an embryo that's developing, here's its eye, its big yolk sac, the rest of its body goes this way. And in this area here is the developing heart. So that's called the pericardial area. And I hope you can see in this bottom image, except for you folks down here, that the pericardial area in this embryo exposed to the stormwater is hugely inflated. And I have a video later, so I won't show you, but the heart is obviously deformed as well. So this is called pericardial edema, and edema is just swelling. Humans experience edema when there's problems, usually with your circulation. And in fact, that's often what it is for the fish as well, is that there's a problem with circulation. Consistent effect that I would see from exposing these fish to stormwater runoff is this small eye phenotype. And this is indicative of a problem with the developing nervous system. So here is a control, so same idea, except now the fish is a little bit older, and the eye is this very large organ taking up most of their head area. In the fish that's been exposed to runoff, the eye is considerably smaller for the same head size. And this figure shows, again, for different dilutions, that even down at 10%, you'll see a significantly smaller eye size. So again, a problem with the developing nervous system. And I have a few videos here just showing some of the other abnormalities, many of them cardiovascular. This first one is not. This is a little difficult to see, but in here, this fish is beating its pectoral fin. So usually at this stage, if it's still inside the chorion, it doesn't really have pectoral fins. But this fish has continued to develop. It's not able to escape the chorion. So this is that delayed hatching. Eventually, if they can't get out, they're quite large inside there and they can't move anymore. And they're permanently deformed if they do escape. So again, this is just one of the many pervasive effects. Some still images here show pooling of blood. So here we have a pooling of blood that would normally be it's trying to get into the heart and then get pumped through the gills and out through the rest of the body. And it's backing up before the heart, pushing into the yolk sac. Here's blood pooling in the trunk area, and this location, in fact, is consistent among different storm events. So there's something particular going on with the vasculature right there. Here's a video, I hope that you can see, of a normal fish. Fish have two-chambered hearts. We have four-chambered hearts, but fish have a much simpler two-chambered heart. It starts as a tube, and then it forms into two chambers, and then those two chambers loop, and they become this twisted shape. And that is required for normal circulation, to have the heart loop. So what I was trying to show here was a picture of a normally pumping two-chambered heart. This, even without knowing what that really looks like, you can probably see that this one is not normal. The blood isn't moving at all here, even though the two chambers are pumping. And also it has some of this cranial hemorrhaging that I was showed in some of those Coho Salmon embryos. And this last video is showing a fish that's got this severe pericardial edema that's swelling around the heart cavity. And the two-chambered heart is not at all looped properly, and this is what results then afterwards, as you get this edema and other abnormalities occur. There's also jaw deformities in that small eye phenotype. All right, so I'm going to move into some of the tools that we're using beyond just these obvious morphological effects. So a really quick primer on genetics. So inside every single one of our cells is the nucleus. Inside the nucleus are our chromosomes. Everybody's heard of chromosomes. Chromosomes are actually made up of DNA that's just all bundled together really tightly. If you unspiral that, eventually you get to a line, let's just think of it as a string of DNA, and along that string is that double helix where all of our genes are wrapped up. So you can think of it as a long string with sequences of genes. Anytime your body needs something done, it sends a message into your cells, into your nucleus, into the right chromosome, locates the gene that it needs along that long string, and what it does is it makes a copy. Opens up the two chains of the helix, opens it up, makes a copy of one of them where that gene is, and that's called transcription. And then it takes that copy and that leaves the nucleus, other molecules attached to that copy, and then start to pump out protein. So that they use the code of that copy to make bring together the building blocks that make a protein. So that's what happens. Gene code gets translated, translation ends up being turned into a protein. And maybe that protein is an enzyme, maybe it's a structural protein that's used to build something in your body. So I have two tools I'm going to talk about just really briefly that I've used for the stormwater research, just to help us get more of an idea about what's going on. So QPCR is one of them, and basically this tells us how much of a gene is being transcribed. How much of this copy is being made of a gene that the body thinks it needs? Is it more or is it less than it should be? And also antibody staining, you probably can't read this, but antibody staining is how much of the protein is being translated. So that tells us how much of your final product that the body is asking to be made, how much is being made. So for QPCR, again, that's how much of a gene is being copied. I've looked at 3 genes so far. One is CYP1A, which it tells us something about contaminant exposure. It's an enzyme involved in detoxification. The other 2 genes that I looked at were related to cardiac assembly. So this one, CMLC2, for example, is involved in actually building the protein of the heart. So one of my storm events, I did 2 separate tests and looked for whether more or less copies of these particular genes were being asked for. And what I found is that both CYP1A was it's called upregulated, so more of these copies were made, so the body was asking perhaps for more detoxification, and more of the CMLC2 was also present. Again, perhaps asking, we need more help putting the heart together. The antibody staining tells us about protein translation, how much of a protein is being produced and where is it being produced. This shows a picture of an embryo, the same picture I showed before with the embryo, so its head is here, this is its big eye, the big yolk sac, and then the rest of its body goes this way. And the green staining, so I was able to send a protein in and attach to the actual enzyme for CYP1A, that detoxification enzyme. It tells me where that enzyme was being produced inside the fish's body. So this shows on the surface that there's a lot of this enzyme, a lot of the detox enzyme being produced in the epidermis of the fish, just in the fish's skin. Obviously, if a fish is being exposed to a contaminant, the skin is going to be one of the first lines of defense. This next picture here in the middle is actually a close-up of just the heart area. So this green line here is that pericardial membrane, and then the yolk sac is right here, but it's black, and then its head is up here. So the red stain in this case is myosin heavy chain, which is just a heart protein, just to identify where the heart is. And then the green is showing where that detox enzyme is. So it's in the skin, like I showed in this figure here, but it's also inside the tissue of the heart. And that is not something you'll see in fish that are not exposed to contaminants that need this detoxification. So on the far right here, I'm showing or as a scientist, we can't help be wondering, why is this happening? What is it in the stormwater that's causing these effects? Now, a lot of stormwater contaminants can be isolated to 3 different sources. The atmosphere, well, you'll have things like phthalates get up in the atmosphere. Automobiles are obviously a big source of contaminants that are going to be in highway runoff, including metals, PAHs, surfactants, and then roads themselves can generate some of the contaminants that we find. Now, as I said earlier, a lot of contaminants that tend to be causing the toxic effects, a lot of that toxicity has been associated with metals, with oil and grease or petroleum hydrocarbons, and sometimes surfactants, which you can think of as kind of like soaps. And in areas where they use salt on the roads to de-ice, that can contribute toxicity as well. Not too surprisingly, too much salt is bad for freshwater wildlife. Now, in the case of metals, we know that metals can be toxic to fish and invertebrates, and it looks like one of the storms that I collected had enough metals to be lethal to invertebrates. So this 114 is January 14th, and it was the one event I collected that was lethal to the invertebrates, but not to the fish. And I'm thinking now it might be because there was so much zinc in the water. So this light blue is the zinc concentration. And across storms, you tend to see a similar pattern. So the most concentrated metal is zinc. The next most concentrated metal is copper. And then some of these are nickel, lead, cadmium, which can also be present, but in much lower amounts. This one storm, however, had a huge amount of zinc and not much else. And this was paired with low DOC, which is dissolved organic carbon. And the dissolved organic carbon can bind up a metal and make it less available. So the fact that zinc was so high and the DOC was so low does make me suspicious that that's what caused the toxicity for the bugs. And I did dilutions of that storm, even down at 5%, that storm was lethal. Are these metals neurotoxic to fish? This has been a really sensitive test that we've employed for a lot of different applications. So with zebrafish, these dots are neuromass. They're places on the fish's body where they have neurons actually sticking right out into the water. And what that tells a fish is when water moves around them, they can feel it. Well, they can feel it, but these are really sensitive cells, these neurons, and they tell the fish information like where the current's coming from, whether a predator is lunging at them. And we have a stain that identifies where these neuromass are and how healthy they are, basically. So they will actually glow green in the dark, which is pretty cool to see. So this is a blow up of one of these neuromass, and these are different hair cells, different neurons sticking right out in an area where you have this highlighted point. So you can count the number of them. And this figure is from a study where we exposed them to dissolved copper, and the copper is neurotoxic. And so at higher and higher copper concentrations, fewer and fewer of these hair cells were present. However, when I did this study with different stormwater runoff events, I found that there was no neurotoxicity. So this is a relative hair cell count at this one site here. So I counted how many of these hair cells were present and then compared it to controls. So value 1 would be the same as the controls. And basically, they were all the same as the control. So I could definitely say that these first flush highway runoff samples, even though they have really high levels of metals, these are copper concentrations were up to 300 in one of these, 300 micrograms per liter in one of these tests. If anybody knows anything about copper, this is really high. However, that DOC I just talked about, the dissolved organic carbon, also tends to be high in these samples, so it helps protect the fish against those neurotoxic effects. All right, so if it's not metals that are maybe causing some of these effects in the fish, what else could it be? We hear about oil and water not mixing, but there are parts of oil that do mix with water. So I don't know if anybody heard all that big news about, Department of Ecology overestimated how much oil was in Puget Sound. If there was that much oil in Puget Sound, you'd see it on the surface. Well, not necessarily, because there are parts of oil that don't just float on the surface. They actually dissolve in the water, just like salt dissolves in the water. This picture I'm showing you is a pH fingerprint. So pHs are one of the classes of chemicals you'll find in something like petroleum oil. So it stands for polycyclic aromatic hydrocarbons, and it's a really big class of chemicals. And this basically lays them all out from naphthalene, which only has 2 carbon rings, and that's the common feature of these PAHs. So from 2 carbon rings up to many carbon rings. So you've got naphthalene and then alkylated versions, so there's alkyl groups stuck to the naphthalene, the dibenzothiophenes, chrysenes, da-da-da-da-da. And this is crude oil, water-accommodated fraction. So it's the part of crude oil that actually dissolves in water. Now, what we know about PAHs is that some of them are relatively safe to aquatic life. So things like naphthalenes tend to be relatively safe. Chrysenes tend to be relatively safe. As far as cardiac effects specifically, I'm talking here. Others of them we know to be cardiotoxic, and they can be cardiotoxic using different mechanisms, which are these different circles I have here. Now, let's contrast the pH fingerprint for crude oil with the pH fingerprint for one of my highway runoff samples. On the y-axis, we're just looking at a distribution of the total pHs. So this is the same layout from low molecular weight to high molecular weight. And what I want to point out here is that you have some of these same cardiotoxic pHs present, particularly what are called the tricyclic pHs. They have 3 of these rings, and we know that they're very cardiotoxic. This pattern in the runoff sample is very reminiscent of the same pattern in the crude oil sample. So pHs could be contributing to this effect. The actual concentrations are within thresholds. A visual example of what I'm talking about, here's a control zebrafish. I hope you're getting used to this. Here's the eye and the yolk sac, and the rest of its body goes that way. And the heart area here. A fish that's been exposed to phenanthrene, which is one of those tricyclic PAHs, has a suite of symptoms. So it includes blood pooled into the yolk area, it includes that small eye phenotype, and swelling of the pericardial cavity. Now, I want to show, again, next to that, some of my fish exposed to highway stormwater runoff. And sorry about the bottom of the screen again, but this one's been exposed to 100% runoff, this one's only been exposed to 50% runoff, but there's similar features, including that small eye phenotype, the pericardial swelling, and also the backed up, the pooled blood. In addition, pHs tend to attenuate quickly in the samples, and this is similar to what happens to the toxicity I showed in that very first slide with where the bugs were exposed. So when I said that I'd stored the sample in the cold, dark storage and then exposed it to the bugs and didn't see any lethality, this is what the actual pHs looked like. So this is micrograms per liter now. So in the fresh runoff sample, and then a week later after cold, dark storage, I mean, they're basically, the pHs are gone. And it's not too surprising when you think that these samples are alive. There's a lot of bacteria in these samples. There's a lot of action going on. It's not just chemicals in water. Similarly, the sample was non-lethal as well.
I have a quick question on that. Is that because, so is it actual biological breakdown, or is it also precipitation of organic carbon, and there's a sediment, it's where a lot of pHs are then held?
So that's a really good question, and You may know that scientists say that when we don't have the answer. But it is a really good question because that's exactly where we need to go to answer this. And I just haven't gone there yet. I'm in touch with a microbiologist, but it's not work I do myself. So right now we don't need to know the answer to that question, but it is very intriguing. All right, so one more thing is line of thought. This is a picture from John in Cardona, who's done a lot of work at NOAA Fisheries looking at effects from oil spills. It was the research that Martha alluded to earlier. So it's the same image I showed you earlier. This is that pericardial membrane. The red is the heart. The green is the detoxification enzyme being activated inside the heart. And you saw this image earlier, but this is one of my stormwater exposed fish right next to the oil, the weathered crude oil exposed fish. So my image is less intense, but it's probably because I took a thinner slice of the fish when I did it. So what I'm pointing out though is these individual cells within the heart, and tracing down through here, this is also heart tissue, just a different layer, so it didn't capture the red. But showing how you have this induction inside the heart, which is not normal unless you've been exposed to something like pHs in something like oil. All right, so, this is where we have reached the downturn apex. Now we're going to go up. It's called the G or something. Yeah. Okay, yeah, I can do that. So we want to know with green stormwater infrastructure, we know that it can reduce metals, we know it can reduce flows, reduce contaminants, but is it going to be enough to prevent these sublethal and lethal effects? So our first step was to look at these high throughput surrogates, and then we're going to focus specifically on salmon and their food webs. And I've just now completed the very first part of this effectiveness research. And rather than jump outside right away in those big outdoor systems that they have down in Puyallup, first we're doing a soil column study. So what's unique about this is that these are large soil columns. Often if there's a soil column study, it's these little benchtop things about this big with soil in it. But these are 14-inch diameter, 3.5, 4 feet tall, packed with the same soil mixtures you'll find in outdoor soil retention systems. Additionally, they had, we were testing the effect of having plants in those soil systems or not having plants. And I'm going to skip the rest of this slide just to get to what we did. So, as you know, it was dry for a really long time in Seattle, and outlying areas as well. I think we all had a similar dry period. Dry for 50 days, and I had a big collection system waiting to catch the runoff, and every day I checked the weather forecast. I'm like, when is it going to rain? I had nightmares like all the time. But it finally rained just a little bit, just enough for us to do this study. I'm not even sure if it was 0.01 inches that fell that day. But we were able to collect in these systems the first flush off of the infamous bridge. And then what we did was we transported all of that runoff in these glass carboys down to Puyallup, down to where we had the green stormwater structures. So 400 liters we transported, and the straight runoff caused 100% mortality in my juvenile coho, which, some of us didn't expect, because we've seen fish at Longfellow Creek, and we're like, they can handle a lot, but this was a lot. 50 days dry is a long, long time. So what we did next was we took the rest of the runoff and we put it through those soil retention systems and had 100% survival. So we're waiting for the water chemistry to come back. There's lots of questions that are still waiting to be answered relative to this. How often can you do this and have it still be effective? But we're going to be learning an awful lot over the next few months.
Were both the plant and the non-planted equally effective?
Well, they both didn't cause mortality, so we're doing a variety of sublethal effects testing as well. So we've sampled tissues from these where we'll be looking for some of those cardiac markers just to show us, all right, well maybe they didn't die, but as you may know, just because a fish isn't dead doesn't mean it won't die or be severely impacted in the future as far as its fitness and survival. So I'm almost at the end here. I have just a very little time left, and I just want to say that our plan is to step this research up and move next year into the large outdoor systems they have down in Puyallup. So these include plots with permeable pavement, rain gardens where they're testing different different plant palettes, and these bioretention mesocosms where they're testing the effect of different soil mixtures Doesn't look like much, so you're not missing much. They have plants in them as well, but they're testing the effect of different proportions of sand and compost and wastewater treatment residuals. So there's a great potential for this research down here because it's unique in the country for being replicated. Often these facilities where they're doing something like this, they have one rain garden or they have one parking lot that they use permeable pavement for, but these have replicated plots, so we're really going to be able to say something sound scientifically. So, in summary, stormwater runoff, as I've been repeatedly saying, is a complex mixture, and it threatens the resiliency of aquatic systems, not just here, but across the country and around the world. We know that runoff can be lethal to developing fish and invertebrates, and that some of the effects that we could expect to see would be cardiovascular toxicity, both in these high throughput screening tests and also in wild fish. I didn't talk about this today, but stormwater may act as an information disruptor because it contains such potent odors. And finally, the green stormwater infrastructure has this great potential to reduce or eliminate these detrimental impacts, and that we need research to help us determine which of these relatively cost-effective solutions are going to be the most biologically effective. So the hope is that even though salmon are suffering this risk of exposure and effects related to stormwater throughout their freshwater life history cycle, with the use of both mitigation and source control and mitigation strategies like green stormwater infrastructure, that we can help this, even though it's not a forest, become more like a forest, act more like a forest. And I don't know that I have time for questions. Someone will tell me. Thank you. Questions? Yes?
In that stormwater treatment, are those columns acting as filters which will eventually just expire, or are they acting as biodegradation? In other words, are they just acting as filters? Or
right, are they filtering
really
or
biologically
right?
Active
And that's
lasts a long time.
Right. So great question, and that's part of the reason we're doing this research is because nobody knows. With metals, we do expect that ultimately these, I'll call them filters in this case, that they're filtering the metals, and at some point they'll reach a capacity where they're no longer able to capture the metals, and metals will start leaching out. So there certainly is an expectation that these systems will have to be replaced eventually, but we don't know when that is. As far as organic contaminants like these PAHs, we don't know yet whether we're getting biological breakdown in those systems or whether, again, it's just a filter, a temporary filter.
How long does the water take to do filtration? Do you use hours and use it, or was it over some period of time?
So I'm not sure if you mean the time between before it got to the filter or after we got it through the filter or the time it took to go through the filter. If I can answer all those. Okay, so we collected the runoff, transported it on ice down to the facility, which took, 45 minutes, depending how fast you drive in the government vehicle. Once we got it down there, we had to mix it, so we poured it into a large mixing tank because each part of the highway maybe was collecting slightly different materials. So we mixed it all together and then started distributing it, collected a side portion that was going to just be the straight runoff, and then the rest of it we put through those soil columns. And it took about an hour to get through the soil columns, perhaps a little bit more, and then we transported it to the fish lab, poured it into different aquaria, took the juvenile sample, we took all sorts of water chemistry measurements and stuff, and samples, and then put the fish in once it got to the right temperature. So the whole thing happened within a half day, I'll say. Anyone?
Probably on it.
Okay, I'll be happy to answer questions later. Thank you.